Hi there, viewers. Having done a video on, two videos, two full videos on cell, I saw a need to release uh, most predicted most possible questions as you can see under cell. And this is for exams such as the for your first year university, so if you're a first year university student or you're um, preparing for the post-UTME or JAMB. These questions are most likely under the topic of cell in biology. Now let's go over them. The first one says that which of the following is considered as the powers of the cell? Very simple question. So the answer to that question is going to be mitochondria, of course, because we've said that the mitochondria are known as the powers of the cell, and that's because they produce ATP. And you remember we said they produce ATP, which stands for what? Adenosine triphosphate. And adenosine triphosphate, I called it the energy currency, right? There's the energy currency of the cell, and it's through a process known as respiration. So via the mechanism of respiration, which follows I think two pathways, and we have discussed that. I've prepared a video on that already, so I'm going to be posting it and linking it in the description below. So we've talked about all of this. So mitochondria performs is known as powers of the cell because it generates energy for the cell. All right. Now the next question two says, which organelle is responsible for protein synthesis? So the organelle that is responsible for protein synthesis or production of protein is the ribosome. We said the ribosome are responsible for protein synthesis because they read the mRNA sequence and they translate it into what the polypeptide chain. We said they are produced in the nucleolus, and they are they 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 contain mRNA which are able to read the the s- the they contain tRNA that can read the mRNA sequence. Is that okay? So they can read the mRNA sequence and produce um, um, they produce polypeptide that is a precursor for proteins, polypeptide chains. Polypeptide are basically um, uh, um, amino acids that are joined with peptide bonds. Amino acids are joined with bonds, and these peptide bonds, when they come together, when these amino acids form large molecules, we call them polypeptides. And these polypeptides can undergo different kind of foldings to uh, become proteins. And I'm going to be releasing a series on proteins as well later on. So the third question says the semi-permeable membrane that surrounds the cell is known as. It is not showing it. It's actually supposed to show the entire options. The the answer is actually between A and B actually, but there are four options here. So I think the screen is not capturing it. The semi-permeable membrane that surrounds a cell is actually known as the plasma membrane. And remember, the cell wall is not semi-permeable, and it's not in animal cell. We said the cell wall was only in plant cell. We said that in that video. So the controlled movement of um, you know in and outward movement of substances, in the cell. That's the plasma membrane. Or the cell membrane. That's another name for it. All right. So let's go to the next question. It says, which of the following structures is found in plant cell but not in the animal cell? That will be the chloroplast. The chloroplast is used for production of food. The site of photosynthesis in the plant cell is the chloroplast. Of course, photosynthesis is the process by which food is manufactured. Yeah, so it's going to be the chloroplast, and it's a plastid. It's a plastid is a membrane-bound organelle, just like the mitochondria. Then the fluid that fills the cell and surrounds the organelle. So the fluid that fills the cell and surround their organ. I remember that we said, now I said we mentioned cytoplasm and we mentioned cytosol. And now these two should not be mistaken. Is that okay? Now you see the cytosol is the fluid. Is that okay? While the cytoplasm is the entire um is um the entire matrix of the cell. So not just the fluid that make up the cytoplasm, there are also cytoskeletons that make up this cytoplasm, that are part of the cytoplasm. But the fluid itself, the fluid content, is the portion of the cytoplasm that surrounds the cell and within the cell rather, and it is the site for most of the metabolic reactions that happen in the. We said the cytoplasm is actually the region, the medium for the metabolic reaction. So metabolism actually takes place within the cytosol, which is the fluid. So this one is the fluid itself, but cytoplasm is the contain the entirety of both the fluid and those cytoskeletons floating within the cell. All right. Then uh, the nucleoplasm is inside the nucleus. All right. So the next set of questions, we have, which organelle is involved in the packaging and secretion of materials? Now, you should um, take note of the word packaging and secretion of materials. Secretion of materials. So that will be the Golgi apparatus. The Golgi apparatus. Excuse me. That will be the Golgi apparatus. The Golgi apparatus is responsible for modifying and sorting and packaging proteins. Is that okay? And lipids, of course. So, and it delivers it. Now, the one thing I mentioned is the fact that it works within the cell. It works within the cell. This guy works without this. I mean, this guy can package stuffs outside, into and out of the cell, while this guy works within the cell. The Golgi apparatus. I would say it was named after Camilo Golgi. In case we come across such question, I'm just reiterating. So Golgi apparatus aids in um, and packaging and modification and sorting of protein of lipids and proteins. All right. So the next is the controller cell. The controller center of the cell containing the genetic material is the nucleus. So the nucleus is the controller center. We call it the brain of the cell. Now, what is the primary function of the rough endoplasmic reticulum? Now, while this guy, Golgi apparatus, is responsible for packaging and secretion of materials, this guy is actually responsible. Now, in this case, this endoplasmic reticulum can also work in packaging of and transportation of materials outside of the cell. But you see, its primary function, because it is the rough endoplasmic reticulum, it means it has what? Ribosome. So because of the presence of ribosome, it's called the rough endoplasmic reticulum. And so it will work for what? Synthesis of proteins. To work for the synthesis of protein synthesis. All right. So, and um, we said that the function of the endoplasmic reticulum is actually, you know, that the ribosome is the one that will produce the protein, but these guys will actually now the ribosome will produce the polypeptide. Is that okay? That's the sequence. This guy will provide the polypeptide, while the endoplasmic reticulum will now give it form and shape. So I remember that a protein cannot function without taking form first. Is that okay? So the form of a protein, in fact, determines its function. Form determine functions, and I'm going to discuss that better when we start the protein, the topic on protein. So just remember that is the function of the endoplasmic reticulum. the protein synthesis. So natural, actually, proteins are synthesized by the ribosome, but they are, given form and shape by the rough endoplasmic reticulum. All right. Next question. Lysosomes are often referred to as, now remember, we said lysosomes or suicidal bags. Suicide, because we said the word lysis. Whenever you hear the word lysis in science, means to split apart, so to split or to cleave. Now, cleavage means cutting. Is that okay? Now, um, this lysis is in the word lyso. Is that okay? Then soma for the from the word means body, such as we see in the word somatic cells, that is cells of the body that grows, and why non-somatic cells of the body that don't grow. But here we're talking about splitting bodies. Is that okay? Bodies that can split. So their suicide bags means they can burst open. Is that okay? So that is another name for the lysosome. And what they do is that they destroy they why they are called suicide bag is because they can actually destroy the cell. When the cell is old, the lysosome is responsible. So take note of that. Then when the cell is old, let me put that down. So for old cells are destroyed by lysosome. Old cells, they are destroyed by what? Lysosome. Right. Then one major function they also perform is they remove waste. Is that okay? And damaged organelles. They remove wastes and other organelles that are damaged in the cell. So the next is that the cell membrane, the main component of the cell membrane is, now, in the cell membrane, we mentioned that the cell membrane, which is the plasma membrane, has um, we called it the phospholipid. Coming, okay. We called it the phospholipid phospho the phospholipid bilayer. All right. So therefore, if you ask that is a phospholipid bilayer, meaning that it has a hydrophobic, hydrophilic, and hydrophilic. What am I spelling? Oh God. It has a hydrophilic head and hydrophobic hydrophobic tail. Right. So what does that mean? It means that the hydrophilic, water loving, is that okay? And hydrophobic tail, water hating, or water hating tail, is that okay? So um hydrophilic. So since it is hydrophobic, it means it's going to be the part that is what? That is the lipid. Is that okay? Lipid fats. They don't dissolve in water, or they don't, um, you know, interact with water. While phosphate group can interact with water. So we call it the um, phospholipid bilayer. So the major components of what will be the lipids. All right. So the next question says, which of the following processes does not require energy? So which of these processes do not require energy? Now the answer is the diffusion. Now diffusion is the only one here that doesn't require energy because it's a passive movement. Remember that these are passive movements. So when now we are probably going to still make, I'm probably going to release a video on that. We have what is called active transport and passive transport. Passive transport. So passive transport. All right. So since molecules division refers to when molecules move from higher concentration to lower concentration, so it requires no energy to do that. It's it's I mean when you in a congesting environment, if a little smell breaks out, it tends to spread fast because it wants to saturate the environment, spread, like it's in a high energy state. It's going to be in a high energy state when they are together, when it's in a higher concentration. So it wants to dissipate that energy. So it works passively, needs no energy for that. And what is endocytosis? You said endo means, while active transport is the one that requires energy. Is that okay? Then endocytosis is endo meaning inside, and um, and cytosis meaning the cell. Is that okay? Now, now it is it is there are two forms by which cell actually there are two forms or two ways by which the cell eats, by which the cell feeds. Is that okay? Now, when the cell folds inwards. Let me clean this so that this can be clear. I just wanted to explain these two other terms so that you understand what they mean. Now, endo meaning inside, as I said, endo, and cytosis refers to the cell. Now, if a cell, there are two ways in which the cell eats. We have the phagocytosis, phagocytosis, and the pinocytosis. Now, when you say the cell is phagocytizing, phagos refers to food, then pinnock here refers to drink. Is that okay? So, um, so phagocytosis is the way by which cell feeds. Why? I mean, when you hear the word phage refer to I think the dead decaying substances or so. But just get the idea that the cell, so when the cell folds inwards to feed, that is called endocytosis. Then, when the cell excretes wastes, so if it folds inward, the cell invaginates around substances. Is that okay? So now invaginate and close up in that substance and digest it. That is endocytosis. While exocytosis um, is the way by which it exudes waste. Exudes waste. Alright, so that's exo, meaning outside, and cytos is still referring to the cell. Alright, the next question says the movement of water across a semi permeable membrane. Now, what is the answer to that? When you see semi permeable membrane, that is going to be what? Osmosis. Once you just see semi permeable membrane, it refers to osmosis. Then, which which type of transport involves the movement of molecules against their concentration gradients? Now, the one that requires the movement of molecules against concentration gradient is called active active transport. So, because it's moving against concentration gradient, so it's going to require energy, like I said in the other time. So, is active transport, movement against concentration gradient. Then, okay, to buttress that point, you know that it is the process of the formation of um, let's use a biomolecule such as okay, for example, the process of formation of ATP, for example, is via active transport. If you remember, that is during photosynthesis now. Okay, I'm going to be releasing the video soon. I've actually worked on it. So, where within the cell there is movement of the hydrogen ions, the hydrogen pump. Okay, I'm going to link the video into the description to explain that part there. So, but the process of just remember that the process of the creation of ATP from ADP is an active transport, and it involves active transport via the hydrogen pump. All right. Then the next question, that will be question 15. Which of the following is not the function? Let me clear this. Which of the following is not a function of the cell membrane? So the one that the cell membrane does not do is we have cell recognition, cell energy production. The cell membrane, okay, does not provide energy. That's the answer. That's the answer. It's the mitochondria that does that. But it does communicate with other cells and transport materials, right? I mean, what that means is that is it, now when we talk about transport of materials, we've talked about the fact that these other vesicles do them. And what they mean here is that those vesicles actually, they don't actually they are the one that package those substances and bring them to the cell membrane. And what they do is that they fuse with the cell membrane and then release those substances outside the cell when they release those substances outside the cell. All right. So that is why cell is actually partaking in transportation of materials. Or that's why we say it is participating in transportation of materials. All right. Question 16 says, "Fac- facilitates facilitated diffusion requires facilitated diffusion." Okay, that will require not ATP, but because if it requires ATP, it means it's using energy. Is that okay? So I think no. The answer actually will be transport proteins. Transport proteins. So facilitated, I mean, transport proteins. So and that's because um, it involves protein that help to move molecules across cell membranes. You understand? That is what facilitated diffusion is. We call them transport proteins. They such as enzymes. Is that okay? Not enzymes. They are just vesicles usually. Vesicles. They not enzymes rather. They are vesicles that just literally carry substances or materials from across the cell membrane without the use of energy. Because if you remember in that video, we discussed when we were discussing the cell membrane, we touched the part of as a part of the cell membrane, part of the makeup of the cell membrane are actually proteins. Is that okay? So some of these proteins are transport proteins. So make sure you see that video to understand this thing better. Now, what is now endocytosis is the process by which cell taking large molecules or particles. We've said that taking large particles or release no, not release, pump ions again no, break break down damage no. So break down damaged organelles. We said the function of lysosome. Is that okay? So the answer is taking large molecules or particles. And the next one is, which of the following is an example of active transport? We have diffusion of oxygen. We have sodium potassium pump. There's supposed to be an hyphen here between the sodium potassium pump, then osmosis of water. So that will be sodium potassium pump anyway. Yeah, I mean, you should know that when you hear active transport, it's actually sodium potassium pump. When that's when sodium ions are pumped out of the cell, and these guys are pumped into like it's called sodium potassium pump because sodium is going out and the potassium is replacing it inside the cell. So that's why they are called sodium potassium pump. And is movement against concentration gradient. So active transport is movement against concentration. We've talked about it. We've seen the question before. So it's movement against concentration gradient. All right. So the next slide we have exocytosis is the process by which that is release material. So the answer is B. Yeah, I think it's already marked there. So the process by which cell releases material outside or expel materials. Um, and they do so in vesicles. I've mentioned that they do so by using vesicles. So it's vesicles that carry this thing that this wastes, and those vesicles will now fuse with the plasma membrane. So that's why we said the plasma membrane is also involved in the process. All right. Then the lipid bilayer of the cell membrane is primarily made up of what? Phospholipids. talked about that primarily. And it also has glycoproteins, but primarily it is composed of phospholipids. And we said it has what? Hydrophilic head and hydrophobic tail. Um, all right. So, and that is why it is selectively permeable. Now, the next question says, which of the following is the correct sequence of stages in cell respiration? Now, we have glycolysis, Krebs cycle, and electron transport chain. Krebs cycle. So the answer is A, because I mean it is Krebs. It is glycolysis, which is the anaerobic portion of that cycle, and then the aerobic is the Krebs cycle that produce high amount of energy, and the electron transport chain. So that is the sequence. And I'm going to be releasing the video soon on respiration. So I've I have a detailed video on respiration, and then I now broke down glycolysis and Krebs cycle. So I'm going to be releasing those videos in quick successions, and make sure that you are subscribed to the channel in order not to miss out on those videos. All right. Um, I said, where does glycolysis occur in the cell? Now it is important to note the locations. In that video, I said that the glycolysis occurs in the cytoplasm, while Krebs cycle takes place in the what? In the mitochondria. So, and in fact, we should know, you be able to you it is important to be able to tell where each process in the Krebs cycle and electron transport chain occurs. Is that okay? So, but for now, let's just answer this question that says what? Glycolysis occur in the cytoplasm, while Krebs cycle occur in the mitochondria. So, and that is where the glucose is being converted to pyruvate. Now, the final electron acceptor in the electron chain. Now, what is the final electron acceptor? I've talked about this in detail in that video. So you should do well to watch that video. I'm going to link it in the description. The, the final electron acceptor in the electron transport chain is actually oxygen. It's oxygen. Now, we said that it combines with electrons and protons to form H2. We said that um, O2 is going to combine with H plus, is that okay? H plus ion to form water, an electron to form water. So then we can balance this reaction by adding our um, number of molecules. So, but this is the empirical state of the reaction. O2 plus H plus, as the proton. I mean now this is, I mean yeah, that's proton. So you could hear proton pump or hydrogen pump. Is that okay? Electron um, to give water. So it's the final electron acceptor, and that also takes place in the mitochondria. In the mitochondria. All right. So then, which of the following? Okay, let me clean this. Which of the following molecules is produced during the Krebs cycle? So which one is produced during the Krebs cycle? Now that is ATP. We know that. That is very simple. That's what is produced during the Krebs cycle. We said how many ATP were produced. We mentioned 36 ATPs produced during the Krebs cycle. Net ATPs are produced during the Krebs cycle. All right. So the next question. 25. Okay. In anaerobic conditions, muscle cells produce what? Very simple question. Lactic acid, right? Well, they produce lactic acid because what? The glucose CH12O6 is not being is not being oxidized. Is that okay? So the muscle cell will produce lactic acid from pyruvate, and um, they will generate NAD plus. Remember that. Or I'm going to link the video in the description. So if you have not seen the video, make sure you watch that video. Um, this NAD plus will allow glycolysis to actually continue. Do you understand? Because it is basically NADH losing hydrogen. So when it loses hydrogen, it becomes, when it loses hydrogen, becomes NAD plus, becomes this plus H plus. So plus H plus, that is on the other end of the equation. Okay, NADH. So NADH. So this guy loses hydrogen and then becomes this. So um that but through that process, this guy C6H12 is converted to lactic acid, right? So from pyruvate, that's C3H6O3, right? So basically, I went over all of these things in that video. So do well to watch the video. The next one says that which of the following organelle, which of the following. So, and in fact, when you see the okay, the other thing is the fact that this lactic acid is actually in the glycolytic pathway. So if you're not even sure of what the answer is, you know that this other one, acetyl CoA, you definitely know that this is going to be the final what will be produced at the end of the day, which is at the end of Krebs cycle, or at the end of Krebs cycle. Alcohol is going to be at the end of anaerobic respiration, um, but the muscle cells actually produce lactic acid. You understand? They actually produce lactic acid. Now, this is a product of fermentation, anaerobic fermentation. So, because we are talking about muscle cells now, that's why the answer is actually lactic acid. So, but these two do not even fit in because they are products after Krebs cycle. Why in this case, we are not even going to proceed to the Krebs cycle at all. All right. So that's another way to just be with. All right. The next one says, which organelle is directly involved in cellular respiration? That will be the mitochondrion, of course, not the nucleus. We know is the powers of the cell. This one produces food. Nucleus, brain of the cell. This one, food, kitchen of the cell. Then ribosome, protein synthesis. So this guy is going to be for respiration. Then the process by which glucose is broken down to produce energy is glucose is broken down to produce energy. Okay, so you see, the question is not hard. It only seem confusing now because you might be tempted to go for fermentation, but actually, the process by which glucose is broken down to produce energy, in this case, would be glycolysis. Glycolysis. Now, glycolysis, the path is the first of the pathways of respiration. Now, yeah, for anaerobic respiration, that will be the only pathway, but for aerobic respiration, that will be the first pathway. Is that okay? So glucose will be broken down, we said, to pyruvate, which is the precursor molecule most of for most of these biochemical reactions, actually. So, and in that video, I also talked about gluconeogenesis, glycogenolysis, just do well to see that video. So but the major thing is that it yields a very small amount of energy, two ATPs actually, two ATPs or net two ATPs. So it's not so large, but at least glucose is still broken down to produce energy. That's the major thing there. So, and it also produces NADH. NADH. All right. The next, okay. During anaerobic During aerobic respiration, the majority of ATP is produced in the mitochondria, of course. That is a very simple question. If you have seen that video, mitochondria. During aerobic respiration, so we said in the cytoplasm, glycolysis takes place. So since glycolysis takes place, it means that only little. We said that this guy generates only two ATPs. It means only little amount of ATP will be generated in glycolysis, which means most of the other amount of this thing required will be generated, as most of the amount of ATP required will be generated via the the um the glycolysis, the Krebs cycle rather, the Krebs cycle. All right, thing is the next one is, what is the primary purpose of fermentation? Now, the primary purpose of fermentation is it to produce glucose? No, because glucose is going to be the precursor molecule for that it's going to be breaking down. Is that okay? So that is the that is so that will not be the primary function of that will not be the primary purpose of fermentation. So is it to regenerate NAD plus for glycolysis? So that is going to likely be the answer. If you remember the process, I mean, for those of you that have seen that video, you will know that we talked about the fact that fermentation regenerates NAD plus from NADH. Is that okay? So that glycolysis can continue when oxygen is, in anaerobic condition. Is that okay? So NADH, NADH will produce NAD plus. So that is the purpose of fermentation. Then, so it is not to produce water or generate oxygen. All right. So which of the following is not a product of cellular respiration? So the one that is not a product of cellular respiration is oxygen, of course. This is produced. I mean, these are the two final products with energy. So oxygen is a reactant, not a product in that process. Oxygen is a reactant, not a product. All right. Then the next set of questions. This one says the process by which cell divides into two daughter cells is called, we have meiosis and mitosis and meiosis. I'm also going to link that video in this video. In fact, I think I have about 2 or 3 videos on that. So make sure you watch all of those videos. I'm going to really be linking in the description because they explain all of these processes very well. And I promise you, you are going to really understand. So make sure you watch all of those videos. Now it says, which one produces two daughter cells? Which process produce two daughter cells? We have meiosis. If you have seen that video, we talked about the fact that meiosis produces four daughter cells. That's why it's called the reductional division. The reductional division. No, actually, there are 2 steps there. So the first step is the one that's called the reductional division, and there's also the equational division, I think. So that is the one. So but overall, it's a reductional division because it uses a diploid cell. It reduces a diploid cell to an aplontic cell. So aplontic. We said the word aplontic is from the word haplus, haplus, which means single. Is that okay? So diplontic means double ploidy. Ploidy number means number of chromosomes set. Including that. Ploidy means number of chromosomes. Number of chromosomes set. So if I say diploid means it has 2 sets of chromosomes. 2 sets of chromosomes. So mitosis is the one that actually divides, that produces two daughter cells, while um, meiosis produces four daughter cells. So it's called reductional division. So four daughter cells means that the number of chromosomes will be haploid, so that if the organism diploid number is, for us, our diploid number 2n is going to be 46. That is our n being 23. So if an organism such as us humans, reproduce our our cells reproduce, it should produce a cell with 2 and 2. So the daughter cells are going to look identical to the parent cell, exactly like the parent cell, while our non-somatic cells. So we said, just well to watch that video because I'm going over many details right now. The non-somatic cells are the ones that engage in meiosis, and the somatic cells are the ones that engage in mitosis. So the non-somatic cells will give rise to apoptotic cells such as the sperm cell, is that okay, or egg cells. So that's that. The answer will be what? The mitosis. Mitosis is going to be the answer. All right, let me try to rush it up. During which phase of the cell cycle does DNA replication occur? During which phase? Now, in this phase of the phases of the, if you don't even watch that video, it means you can't comprehend any part of this. Um, the phase we have the I said there is the S phase, the G1, S and G2 phase. So this G1 stands for gap phase. S stands for just check that video in the description. So, but the phase where the DNA replication actually occur is the S phase. Is that okay? So that S stands for synthesis. Is that okay? Synthesis, yes. So G1 will stand for gap, G2 will stand for gap 2, gap 1 and gap 2. So more like that, like is that phase, those first two phases, gap 1 and gap 2 phases are phases of rest, kind of. Is that okay? So these three make up what we call the interface. So that was why when I introduced that video, I told you that interface is not a part of the cell division stage. It's not a part of the cell division phase. So what it does is that the what this phase does is that it makes sure that we have DNA that is enough to recreate daughter cells that will receive equal number of chromosome sets that will have the same ploidy number. Is that okay? So the answer is S phase here. All right, let me change this. Okay. So the next one says, which of the following occurs during prophase of mitosis? So the one that occurs during prophase of mitosis, we have chromosome aligned at the equator prophase. So remember that prophase, we have the PMAT. Okay. So PMAT, P being the first phase. So is it that chromosomes align at the equator? Right. We know that that is wrong because at the equator, that is middle, right? Middle. And I said middle stand for what? The metaphase. So chromosome condense and become visible. Okay, that's more likely to be it. But maybe something might actually occur before that. Let's see. Sister chromatids separate. No, as a way. Okay, so the answer is B. Chromosome condense and become more visible. You know, prophase I said is the longest. So remember that also is the longest phase of the cell division. So and and it marks the beginning. Is that okay? I mean, that's just it. But view the video, check the video. I keep talking about that because I, broke down all of these concepts in those videos. All right. What is the result of meiosis? We mentioned that four haploid. Yes, four haploid. So that will be D, option D. Four haploid daughter cells. While the product of mitosis will be two diploid cells. Now, the exchange of genetic material between homologous chromosomes during meiosis is known as. Now, there's a process known as recombination. So it has two names. So in case you were, doesn't it has two names. We have what is called recombination or crossing over. Crossing over. Now, this is a process that crossing over. Now, this is the process that makes variation possible. Now, I'm going to be releasing a video on evolution soon. So that video I'm going to be talking about the concept of variation, but still see the video, the content of the genetic video that I released. Just type on, okay, maybe I'll link it in the description also, but it's a very long time I made the video. So I talked about concept of variation. So it is crossing over that actually makes it possible for for variation to be possible in organisms. So you know, um, when it says when homologous chromosomes when they pair up, so that's basically just them, the two chromosomes coming together. So when they separate, they will separate as non-sister chromatids, non-sister. But when they were separating, if it were to be in mitosis, when they will be separating, they'll be separating as sister chromatids. So we say sister chromatids separate, that's the language you use. But in this case of meiosis, we say non-sister chromatids because the chromatids that will separate in this case are not, in fact, they are not chromatids that actually separate in this case because they are chromosomes that separate. And I said there's a difference in chromosome and chromatid. I can't get into that now in order to not make the video too long. So see that video in the description. So the answer is going to be crossing over. Which structure holds sister chromatids? Okay, I didn't mark this. Holds sister chromatids together. The structure that holds sister chromatids together is going to be what? At the center, isn't it? That's central. Is that okay? Centromere. Centromere. Spindle fiber is that fiber that is formed by what we call, do you remember I called it the kinetochore? Kinetochore. All right. So then the telomeric region is just the end of the chromosomes. So when chromosomes are held at the telomere, we call it telomeric um, pair. Telomeric chromosome. Is that okay? We have no telocentric rather. No, not telomeric. I'm sorry, telocentric. So telomere means the end, the tail portion of the chromosome. So when chromosomes are held together at those points, it's still this portion will still be called the when they are held together at those points, such as we have this. So we call this telocentric chromosomes. Telocentric, and we have metacentric and subtelocentric and submetacentric. Right. I'm just getting into too many things. But let's just continue. Just make sure you are with me because even with this, you are going to get as much as possible information. But make sure you see the videos I link in all the videos I link in the description in order to get a full overview of what I've been discussing. During which phase of mitosis does the sister chromatids separate? So separation is, I said in that video that P stands for prepare, M stands for middle, and A stands for away. So that will be away, that is anaphase. All right. The next one. What is the significance of GO phase? GO phase in the cell cycle. The GO phase. Now, the GO phase is when the cell. Excuse me. I'm trying to charge my phone. Okay, what is the significance of the GO phase in the cell cycle? Now, the GO phase is the first phase where the cell, I said, is the gap one phase. So, it's also called the GO phase. So, it's where the cell actually rests. So, or like that's the terminology that is being used for it, that the cell is resting. Is that okay? And that is when it stops dividing. That's just naturally what it does. Is that okay? So, and some cell can actually remain in this phase forever. So, such as the nerve cells. I mean, before they die, that's what I mean. Forever. Is that okay? So, what is the significance of this phase? Is it DNA that is replicated? No, we said that one happens in the S phase. Cell permanently exists in the cell cycle. Okay, 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 okay. So nothing is being said of the arresting here. So it's in the G2 that is prepares for for division. So in this case, the answer is going to be the cell permanently exits the cell cycle. Okay, yes, it's okay. It permanently exits the cell cycle to rest. So that's still connoting rest. It's okay. Which of the following is true of cancer cells? Number one, they undergo normal cell division. That is very, very untrue. So they have, in fact, it's the opposite that is true. They undergo uncontrolled cell division. All right, that will be the answer for that. Let's go to the next one. The next one says, uh, the phase of the cell cycle where the cell spends most of its time is what? Is it the M phase, S phase, G1 phase, G2 phase? That is actually the G1 phase. So it's the first phase of the cell cycle. That is the way it grows and carries out normal function. So, oh, okay. Yeah, I was supposed to actually talk about that. Sorry. The G1 phase, there is G1, G2, as I said. So that is G1, G2. That those ones are the phases that are, they are we call them the interfaces. That okay during cell division. But before this is the GO phase, G0 phase, where the cell actually rests. Is that okay? Right. So then here we have the cell communicates with each other using what? Is it DNA? No. Is it enzymes? No. Now, because now some people might be tempted to say enzymes because of the fact that they are enzymes that the cell, you know, that go within and out of the cell. But the cell actually use chemical signaling. And I mentioned that when we're talking about um the cell membrane, yes, the cell membrane. That it has some molecules, peptidoglycans, um, glycoproteins that it uses to communicate, do you understand, with other cell, you know, and they trigger a number of molecular reactions, that will create, you know, some of these signals, like they send some of these signals using hormones, using neurotransmitters, So, and those things, they target those things are for targeted cells. Is that okay? That was what I discussed in the cell membrane. I talked about the fact that these neurotransmitters and these molecules are they are they are they have specific receptors on different organelles that they are communicating with. Do you understand? It's just like like a slot where only one type of card can fit into. So, right. In the process of receiving and responding to chemical signals is known as what? Now that is also very simple. When cell receive you know and respond to chemical signal, that is called signal transduction. Signal transduction. Right. We know what replication is and we know what metabolism is. So those are not. So the answer is cell transduction. So and it triggers reactions when the cell sends signals. It's going to si- you know lead to a reaction of the other cell. All right, because of longevity, I'm going to stop the video here. If you want the full slide of the complete slide here, you can reach me on WhatsApp via the number 09021416307. I think I wrote that well. Oh God, cleaned everything again. 09021416307. So, or you can just write +234902141 da da da. So you can reach me. I'm going to send the full slide of this for you. And I also have a set of more than 300 questions, an archive of 300 questions. It comes at a very small cost anyway. 300 questions um, on both of cell and everything that pertains with cell, everything that pertains with cell, both and it starts from the thing about this archive is that this set of questions that they go from relatively simple, not relatively, not so simple, is that okay? For at least for an average biology, not an average biology student, a student in the that has just finished secondary school to be able to attempt all of these questions. Those are the simple stage up to the advanced stage on that cell. So I have this 300 questions. So and I have another one where and I they have answers. Is that okay? I'm going to provide the answers for you. I'm going to provide the answers, but I have their questions in my archive. So these questions in my archive. So do well to reach out to me on WhatsApp. And that'll be it for this video. If you have not already, do well to subscribe to the channel and click the bell notification so that you'll be notified whenever I drop a new video. So that's why you should actually subscribe to the channel because I drop contents like this so that you can have access. I mean, so that you can and also do questions. There are not so much platform that does that. Is that okay? So try to subscribe to the channel so that you don't miss out on any of these things. Try to subscribe to the channel. All right. See you in the other videos. Thank you.